Morning. Good morning. I'm Paul Howard, the Director of Public Policy at Amicus Therapeutics. On behalf of my colleagues at the Penn Orphan Disease Center and Amicus, I'd like to thank everyone for joining us for our third webinar session on harnessing non-proprietary patient registries and real-world evidence to accelerate rare disease drug development. While our first session focused on the FDA's role in defining regulatory frameworks for evidence collection and use cases for RWE that can help align the community on shared data standards. And the second session reviewed best-in-class rare disease patient registries that can help us bridge the gap between drug development and clinical care. Today's section moves a little farther afield and looks at applications from the oncology community, asking what we can learn from the rapid advances in cancer treatment, including targeted therapeutics, immune modulating drugs, and CAR-T, that have helped transform cancer treatment for many malignancies over the last decade. In many ways, oncology has become the paradigm for rapid regulatory science innovation, with industry, FDA, and patient groups working closely together to solve critical translational science challenges, especially related to applications of real-world evidence. There are some important similarities and certainly some differences between oncology and rare diseases. But what they share is that cancer is also a highly fragmented genomic disease, with sometimes dramatic variability in patient progression and response even in the same disease state. But thanks to leadership from key stakeholders, including the U.S. Food and Drug Administration, the oncology community has managed to standardize key data elements and concepts to help solve previously daunting technical challenges and personalize cancer treatments that have made a real and often dramatic difference in cancer patient outcomes. These gains have been made possible by a powerful synergy between next-generation sequencing platforms and precision medicines that can be used as probes to test how well researchers and clinicians understand the critical biological pathways involved in cancer growth and metastasis. Today's panel will focus on how these key partnerships and lessons learned evolved in areas like real-world endpoints, novel biomarker development, and innovative clinical trial design. Our chief provocateur for this session is Jonathan Hirsch, the founder and president of SciAps, a market leader in precision medicine and real-world evidence solutions. John founded SciAps in 2008, and he's been one of the pivotal leaders helping to advance the field of precision oncology at key inflection points, including catalyzing national cancer data sharing networks through the White House's Cancer Moonshot Data Sharing Working Group, and working closely with the FDA's Oncology Center of Excellence and FDA leaders like Richard Pazder and Amy Abernethy to validate new approaches to the collection and analysis of real-world data, helping drug developers, patient groups, and industry partners turn data into evidence using cutting-edge analytics that can help inform regulatory decision-making and patient care in the real world. Please join me in welcoming, virtually, John Hirsch.
Thank you so much, Paul, and thank you to the Amicus teams and Penn teams for having us today for this very important event. As Paul mentioned, my name is Jonathan Hirsch. I'm the founder and president of SciAps. Back in 2008, when we started working on SciAps, the concept of bringing together real-world molecular data, clinical data, treatment, and outcome information to try to assemble a national knowledge corpus to advance precision medicine in oncology was not just a novel concept, but was seen as too far ahead, as too boundary pushing, as something that was too difficult and probably would never happen. Now, 10 years later, it's been a very long road, but this has come into being through the efforts that we and many other stakeholders in the United States and internationally have pushed forward. I've been very pleased to work over the past few years with a number of the folks on the panel today, and I'm looking forward to hearing all of their perspectives on how we can bring the lessons learned from a decade in oncology into being for rare diseases. Next slide, please. Over the past decade, a number of the folks on the phone, as well as various partners, work together to bring real world evidence to the forefront in oncology. This has really been a collaborative effort across a number of different stakeholders. There's been the National Cancer Institute, the Food and Drug Administration, pharmaceutical companies, molecular testing labs, academic medical centers, and various other stakeholders who have all come together to make this happen. I chose a few specific events to call out as an exemplar of the decade of progress that we've made. The first one being the creation of a knowledge map to help understand the molecular landscape of oncology, which was first and foremost promoted by the Cancer Genome Atlas effort through the National Cancer Institute. A second component of progress has been the use of real-world evidence to advance molecularly targeted therapeutics in the regulatory setting, in particular the Amgen Blincyto example that we'll talk a little bit more about. Next was the efforts promoted by the federal government in the form of the White House Cancer Moonshot to advance knowledge sharing and knowledge networks across the United States. Then we had 21st Century Cures, which really formed a framework for data sharing and collaboration, in particular promoting the establishment of rules of the road to allow for the sharing of clinical data across multiple institutions, but also promoting the FDA in creating a framework for the use of real-world evidence in a regulatory setting. That then resulted in the creation of the real-world evidence framework that we'll talk a little bit more about today, that the FDA put out in 2017. And then a number of different organizations, including Forensic Cancer Research, who promoted cross-industry collaboration efforts to bring together multiple different stakeholders to validate and advance the use of real-world evidence, not just in a regulatory setting, but in settings beyond that. Next slide, please. As I mentioned previously, a lot of these efforts rooted in TCGA, which really started in the mid-2000s and gained a lot of steam in the 2010 to 2013 timeframe. One of the most famous examples are the two figures on the left side of the page, which laid out a molecular landscape of various tumors and really helped highlight to the industry that cancer was not just a collection of individual tumor types, but really a very broad collection of molecularly driven aberrations that happen to cluster in particular anatomical locations. And this was a sea change in the oncology domain that really helped start redefining cancer as a collection of many different molecularly driven rare diseases rather than tumor type of origin driven diseases exclusively. And this really helped promote the thinking in the oncology domain that in order to gain a handle on oncology and therapeutic development, we needed to really start bringing together data from across many different silos to assemble a larger and larger data set study. On the right side, you see one example of this effort put into real practice, which was the effort out of the University of Wisconsin and the Aurora Cancer Center teams to look at the real-world utilization of molecular testing in clinical practice and understand the therapeutic options given to patients post-molecular testing and what those patients' outcomes were. So this was one of many efforts to collect real-world treatment and outcomes information driven based on molecular profiling data and use that to begin sub-segmenting patients into responders and non-responders based on their molecular profile. So this and many other efforts that emerged out of it were really rooted in the foundations of the data sharing networks created as part of TCGA. Next slide, please. As mentioned previously, following the passage of 21st Century Cures, the FDA and specifically the Oncology Center of Excellence really took the lead in helping to establish a real-world evidence program. And of course, we'll hear today more from Guidon Blumenthal and Jeff Allen on this topic. But the real-world evidence program really helped to create a framework for organizations who were thinking about using real-world evidence in a regulatory setting and bringing this to the agency. Next slide, please. The FDA outlined very clearly for a number of stakeholders, not just in the pharmaceutical industry, but the stakeholders who were building registries and other real-world data efforts, the various different uses of real-world data that the FDA expected to see in the regulatory setting. And you can see at the bottom the outline of the increased reliance on RWD throughout the drug development and regulatory life cycle. Next slide, please. As part of this outline, the FDA really called out the various uses, in particular of single-arm studies using external controls and featuring this as a particular example of interest. And as you can see at the bottom, specifically calling out not just oncology, but also applying this to rare diseases and the expectation that various stakeholders, whether it's pharmaceutical companies, real-world data companies, or the two in combination, bring these sorts of examples and use cases to the FDA for collaborative work. Next slide, please. One of the many organizations that really stepped up and took a leadership position in this and deserves to be called out for it is Amgen, and you'll hear a little bit from Chris today about this example. But Amgen was really a trailblazer in Blincyto in using historical real-world data to form an external control and submitting that to the agency as part of a regulatory approval for Blincyto. And you can see two of the articles on the left side of the page, and then on the right side of the page, a presentation on the replication of the overall survival from the historical comparator to the subsequent phase 3 clinical trial. And Chris, I'm sure we'll go into more details on this today. Next slide, please. Following a lot of this development and movement, several components of the FDA, in particular the Oncology Center of Excellence, as well as the pharmacology group, published commentaries and perspectives highlighting the various uses of real-world data and real-world evidence in the regulatory setting. So on the right side in particular, you can see the publication by Guidon Blumenthal and his colleagues at OCE that highlights the uses in oncology of real-world data. Next slide, please. These articles, when you take them in aggregate, really called out 3 particular high-profile regulatory use cases. The first use case was characterization of real-world populations. So in particular, using real-world data to form a more comprehensive understanding of patient journeys to generate insights into testing patterns and treatment patterns. Now in oncology, this has been particularly important in order to understand the utilization of molecular testing and how this has impacted patient outcomes as molecular testing has become more and more used. The second, which is a topic that we'll get into quite a bit today, is the understanding and validation of endpoints, or as we've now termed them, real-world endpoints, to understand response to therapy. And the FDA and OCE has taken in particular a strong leadership role, and you'll hear more from Guidon about that today, in understanding how to validate real-world endpoints and how those endpoints may be distinct from traditional clinical trial endpoints. And then the final strong position is safety and dosing insights. So one of the areas in which real-world data can be particularly helpful is using the much larger populations that real-world data can represent to pick up safety signals, pick up dose modification signals, and understand how that might be used to update labeling after the therapy is approved. Next slide, please. Now, many organizations have picked up the mantle, and in particular, Friends of Cancer Research has done work over the past several years to pull together many stakeholders who otherwise may be competing. So organizations such as SciAps, Flatiron, Tempus, Concert AI, CancerLink, many organizations who might be competing in the market. Friends of Cancer Research has actually managed to pull these organizations together to work collaboratively in a pre-competitive manner to establish rules of the road around utilization of real-world evidence. And what these efforts have done is really demonstrated the capability to mature the real-world evidence space and to work together to advance things like data standards, analytical standards, how results are presented and communicated, and how the agency engages with organizations to understand the utility of their data and their analysis with respect to any other organization. So we'll hear more from Jeff about these efforts today. Next slide, please. And that brings us to our panel. So today we have a fantastic group of panelists who have all touched this space directly over the past decade. And today we're going to be talking about how has and why has oncology made such rapid progress over the past 10 years? What has been the role of the federal government, and in particular the FDA OCE, in advancing these efforts? And what role have collaborative efforts such as Friends of Cancer Research played? How can we use a use case driven methodology from oncology to develop and advance data standards, and in particular working on endpoint validation? And then can we apply any of the lessons from precision oncology, so the subsetting of cancers into very specific rare diseases, can we apply that to the rare disease? So with that, I'm going to introduce our first panelist, Don Blumenthal, who formerly was of FDA OCE and is now the VP of Global Regulatory Affairs at Merck. Don?
Yeah, thanks, John, and great to be here today with you all to discuss this great topic. So as John mentioned, I'm currently VP for Global Oncology Regulatory Affairs at Merck. I joined Merck about 7 months ago in February of this year, right before the lockdown. So I was able to meet several of my colleagues in person, and now we're in a remote environment as everyone else is. As John had mentioned, I was previously at the FDA for over a decade. I came out of my fellowship at the National Cancer Institute in Hematology Oncology, and was a clinical reviewer focusing on mainly on breast cancer products, then a clinical team leader in thoracic and head and neck cancer. I was an associate director in precision oncology in the immediate office of oncology office. And then, as John alluded to, with the 21st Century Cures legislation and the cancer moonshot, that enabled the mandate to create centers of excellence within the FDA. And the first center of excellence and only disease-specific center of excellence is the Oncology Center of Excellence. And I had the privilege of helping to stand up that organization under the leadership of Rick Pazder. I'm a hematologist oncologist by training, and my focus has been mainly on thoracic malignancy. Next slide. So I agree with a lot of what John mentioned in his opening remarks. I think if you look back at the last decade in oncology, it's been quite remarkable in terms of improving outcomes for patients with cancer, both common cancers, broadly speaking, like lung cancer and breast cancer, but also, with for patients with rare forms of cancer, and molecular subsets of common cancers. And this is due to better understanding of the underlying molecular pathogenesis of cancer, better, smarter, faster, cheaper diagnostic tools like next generation sequencing, and more rational drug development, better drugs through improved science. We've also seen tools from Congress, including tools that FDA Oncology uses frequently, such as accelerated approval and breakthrough therapy designation, which has helped the cycle of innovation. And we've seen, an increased tolerance of risk and innovation from industry. And FDA has kind of led the way in providing guidance for industry, and these include, for example, expansion cohorts from first-in-human trials, master protocols such as basket trials and umbrella trials, and adaptive clinical trial designs, including platform trials. We've seen, following the lead from the HIV community, very strong patient advocacy in oncology, and that has brought sort of this sense of urgency that we can't wait for perfect data. We can't always wait for overall survival in 2 randomized controlled trials with p-values of less than 0.05 in every circumstance. We have to rely on some uncertainty, and I think that has served us well. And then, of course, as John also alluded to, and Paul in his opening remarks, we've seen very strong visionary leadership in oncology under Rick Pazder, who's very willing to always keep the patient as sort of the North Star in all of this, and focus on delivering what's best for patients in terms of better therapies. So we can go to the next slide. And just a couple of thoughts on, what we might need to do in the next 10 years. In oncology, John's company, a lot of other companies are looking at ways to better incorporate electronic health record data and other data to support prospective clinical trials, as well as using observational cohorts. And some of the use cases that John alluded to in terms of characterizing your patient population, focusing on endpoints, evaluating safety and dosing in the real world, have already been used quite extensively in many NDA and BLA submission packages, and will only increase in the future. In terms of real-world endpoints, I think we'll have a lot of further discussion on that. I think that oncology has been served well by using objective response rate and durability of response of sufficient magnitude in single-arm trials for approval decisions. I think we've been fortunate. Many other rare diseases may not have the benefit of having these type of measures that are objective, and when they're observed in sufficient magnitude, can be clinically meaningful in and of themselves for patients. But that being said, we're going to have to work on, and the work is already being done through, organizations like Friends of Cancer Research, which Jeff will discuss further on implementing further real-world endpoints. And then, one area, that we'll have to think about coming down the road are emerging biomarkers like circulating tumor DNA changes, pathologic response, novel imaging modalities like radiomics. It's going to be hard to validate associations, particularly in rare cancer types. And we're going to have to use kind of the same approach or a similar approach like we did with tissue agnostic drug development, where you're borrowing information across histologies. We may have to use a similar approach when we're validating some of these novel endpoints. So I think I'll stop there. I look forward to the discussion, and thank you.
Thanks so much, Gidon. Next, I want to move to Christopher Kim, who is the Director for Observational Research within the Amgen C4 group. Chris?
All right, thanks, John. Hi, everyone. So my name is Christopher Kim. I'm a cancer epidemiologist by training, and I'm a director in Amgen in the Center for Observational Research. I lead a team in developing relevant studies to support our medicines in the hematologic malignancies area. As John mentioned, I worked quite a bit in the Blincyto program, blinatumomab going forward, and was quite active in the development of novel medicines for other acute leukemias, multiple myeloma, as well as lymphomas. We worked on studies, as simple as identifying the treatment landscape changes over time in multiple myeloma, to studies as complicated as using a propensity score adjustment to compare phase 2 study data to an external control with Bayesian augmentation to improve statistical power of two populations with less than 50 patients in it. Talking about the second bullet point here, some of my relevant experiences. As I mentioned, we worked quite extensively in the blinatumomab program, where we used real-world data and non-trial data to help lead to initial approvals and label expansion. I worked pretty closely, in particular, on the label expansion for the minimal residual disease expansion, which was the subject of an advisory oncology drug advisory committee, as well as the EMA discussions about the Philadelphia chromosome label expansion. These papers now, where we use external data to compare to single and trial data, have been published, where we use innovative methods, and those are available, and we can talk about those during the discussion section. Additionally, we've also used multiple databases to satisfy post-marketing requirement studies, which evaluated potential safety signals that may have been associated with drugs. In our case, that was a carfilzomib program for the treatment of multiple myeloma. And then lastly, we're also working with the FDA on a potential pediatric acute leukemia study where these patients have no other options left and are in dire need of additional treatment options, particularly for the treatment of T-cell acute lymphoblastic leukemia, a very rare subtype after relapse, where there have been very few innovative medicines approved since 2007. And this approach potentially could help enable a new novel therapy combination to be used for this patient population. What is really critical in the use of real-world data are really appropriate study designs, data that are really fit for purpose. This includes understanding that the data must have relevant time periods, patients must come from the same source population, and you must use appropriate statistical analysis. As more and more real-world data really become available for use, the appropriate epidemiologic design principles need to be implemented within the framework of causal inference for this data to become relevant real-world evidence, which then can help enable appropriate decision-making. So as an expert in this area of epidemiologic design, I'm hoping to contribute further to this area of real-world evidence as appropriate, and looking forward to the discussion today.
Great. Thanks so much, Chris. Next, I want to introduce Allison Campbell. Allison is currently the head of digital health oncology R&D at AstraZeneca, having joined during the pandemic. and previously was at Roche Genentech, where she led patient-reported outcomes group. And one of the things that I think is particularly interesting that Allison, I'm going to prompt you on is the intersection of real-world data and patient-reported outcomes. So take it away.
Oh, great. Thanks, John. So it's a real pleasure to have an opportunity to discuss this today, because I think there's an enormous amount of great analogs in the oncology world as well as in hemophilia that can be applied through real-world evidence to increase the insights we have, to have improved endpoints, and with better endpoints, we can better develop drugs that will benefit patients. You can go to the next slide. There's just an example there. So in my experience, I like to think about endpoints. So what's the concept of interest that's most relevant to a patient? And how do we measure that best, whether that's through a device, whether that's through creating a software as a medical device model, whether that's through digital symptoms, and then how do we analyze that in a rigorous and reliable way that the agency would have confidence to communicate that in labeling? Because if we're collecting data that's not accessible to patients and their families for decision making, then I think that's a missed opportunity, because that really is our end stakeholder. One piece I'd like to highlight is I think pre-competitive collaborations, and I'm so glad that Jeff Allen is here today, have really enabled oncology to move forward so quickly. Most of the folks on this call I've gotten to know through work with Friends of Cancer Research, longevity, and academic collaborations. An example I wanted to highlight is I'm part of the CISAQUAL group, which is setting international standards in the analysis of quality of life endpoint. And what was great about this group was there was so much heterogeneity in how this data was collected and analyzed that it wasn't credible to the external community. And by coming together through consensus work with papers published in the Lancet Oncology, including regulators, global regulators, so members of OCE at the FDA, members of the EMA, the academic community, and the industry community, we were able to set standards that, when systematically applied, allow us to aggregate insights across trials. And so the reason I think this is really important for real-world data is that we have this opportunity to not align on how we measure it. We're always going to have a different EMR. I mean, within the US, we have so many EMR systems. I build digital therapeutics as part of my current role, and the interplay and interface into those systems is complex. And in the EU, obviously, they're different as well. But if we can align on what's important to measure, when to measure it, then we can allow those individual systems to collect the data and whatever makes the most sense for them. But if those core concepts are aligned, we can then start aggregating them for rare disease and deriving insights. And cell and gene therapy is a great example of this as well. And so I'm really excited to be part of this conversation, because I do think the more we can align on aggregating information to have better endpoints and targets, the more that industry can develop drugs that are going to most benefit patients on the concepts most relevant to them in rare disease.
Great. Thank you so much, Allison. And finally, I want to introduce Jeff Allen, who is the president and CEO of Forensic Cancer Research. Jeff?
Great. Thanks, John. Good morning, everyone. I'm Jeff Allen. As John said, I'm with a group called Friends of Cancer Research. I've been there for about 14 years, so while I happen to be a molecular biologist by training, I won't necessarily dwell on that. I think if you jump to the next slide, I provided a little bit of information about what my organization has been working on over the last couple of years. And if we don't have that slide, then I can probably just go from memory. Oh, there we go. So our organization, as mentioned, is really driven by the collaborations that we're fortunate to be part of and have been for the last number of years. And I really want to express my gratitude to everyone who has partnered with us on a diverse set of topics over the years, because the work would not have been possible without people's willingness to come together and think about doing things differently, and actually in a collaborative way, putting perhaps even competitive nature aside in order to work on things that essentially can be a rising tide to lift all boats. As you can see depicted in this picture here, our initiatives range across a diversity of different settings, including things like educational programs that we work with a number of different organizations that have broad outreach, policy organizations to think about ways that cutting edge science can inform legislation and policy initiatives, also done in a collaborative fashion, as well as initiatives related to science and advocacy about doing things differently. I've listed here a couple of examples of that, including the Lung Map Master Protocol, which was on the earlier side of personalized medicine collaborations, bringing together multiple different companies under a common screening protocol in order to better identify patients in a biomarker-driven way and provide, at that time, accessibility to molecular testing to help guide the potential treatment options that they had available to them through this collaborative clinical trial, which is still running now 6 years later as one of the largest personalized medicine trials implemented in large part through the NCI cooperative groups, as well as the numerous pharmaceutical companies that have regularly contributed new drugs in order to keep this mechanism moving. And also an initiative here around looking at harmonizing different approaches toward validating biomarkers around, in this case, tumor mutational burden, with the understanding as science moved forward that increased mutational levels within certain types of tumors may be an indicator that they could be more responsive to immunotherapy. But in looking at ways that mutational burden could be assessed, it was quite diverse. So to try and provide a uniform way for reporting and counting mutations and really setting up a research strategy across numerous different investigators and companies enabled things like the pursuit of agnostic type clinical trials in order to test using mutational burden, test the potential use of various IO agents in numerous different cancer subtypes simultaneously. And I raise those just as examples, but perhaps more relevant for today's discussion has been a real-world evidence partnership that we have been participating in over the last couple of years that John mentioned, that has been a collaboration with 10 leading healthcare companies, including his, alongside with FDA, NCI, numerous drug developers, and other patient advocates, to establish a pre-competitive learning platform looking at the potential utility of real-world endpoints. What we tried to do by bringing numerous different data sets together was to work on things such as developing a common set of definitions and various data elements that could be deployed using real-world evidence, explore different methodological approaches using real-world data, and ultimately run parallel analyses in order to answer a common question. What we hope that this would achieve by doing this multiple times over, albeit on independent different data sets, was to try and identify key variables that could lead to the differences in the observations that each data participant was able to bring back to the partnership and share about their learnings. So in this case, the key question was centered around advanced non-small cell lung cancer in patients that were treated with immune checkpoint inhibitors. And through the first set of pilots, we explored the ability to create uniform standards across different data sources and assess the ability to extract endpoints on or extract information on different endpoints. Typically in oncology, we rely heavily on endpoints like overall survival or tumor measures such as progression or response. Those can be difficult to measure using electronic health data. So our collaborators helped design a series of experiments to look at things like time to next treatment or time to treatment discontinuation as potential indicators of response to therapy. And what we've just been in the process of finalizing and releasing has been looking at the extent to which those endpoints are able to be used to distinguish between different therapeutic options in the post-market space in order to continue learning once products are on the market and better characterize how they perform over time. So I raise that as, yes, it's been a very interesting and terrific collaboration in the oncology space, but perhaps that's a starting point for our discussions where these types of prototypes could be helpful to inform other collaborative efforts in large populations such as COVID-19 or in small populations that we'll be discussing today, like rare diseases. So I appreciate the opportunity to be here and join the discussion.
Great, thank you so much. So moving into the panel discussion, the first item that I wanted to tackle head on, because I think it's always a discussion in these sorts of settings is, as we reflect on the past, say, decade of progress in making real world evidence a more routine part of our cancer research, development, and care journey, how did we make so much progress past 10 years? And in particular, what was the role of the FDA, and not to toot the horns of the folks on the phone, but of Forensic Cancer Research and other collaborative efforts? What was the role of those organizations in helping to promote and push forward progress in this area? And as we think about our past decade of progress, do we think that this progress would have been possible without the leadership role that the FDA played? So I'll toss it out to whoever wants to start. And if not, I'm going to call on someone.
Well, I'm happy to start. I think that the FDA has played a very unique role in driving these oncology collaborations through their participation, their vision, their leadership, and the willingness to work with the external community. I think it's been very remarkable to see over the better part of a decade or more how the Oncology Center of Excellence has really been formed on the foundation of being a part of the solution. Yes, they play a role, absolutely, of the regulator evaluating the validity of data that's brought to them. But I think there's been a recognition across the oncology enterprise that we're all in this together. And by existing in artificial silos where they're not necessary is not helping anyone. And I think FDA has done just a remarkable job of leading the way and bringing those silos down in a way that has made the process much more collaborative. And it should be of no surprise that in a field where science is evolving so rapidly, everyone is probably identifying very similar solutions, and we're going to be far better off trying to solve them together than separately.
I think some additional factors that really contribute. One is actually really an epidemiologic phenomenon in that as we live longer and longer, there's just simply more people that get touched by someone who has cancer or has seen someone pass away from cancer. Cancer is increasingly more common, and cancers are often very fatal. When you have that kind of contribution and impact to people's lives, you just get more interest overall. And it makes, what Alice has mentioned quite a bit about endpoints. If you're looking at something like overall survival, that's a very objective thing to see. It makes it very ripe for trying to see what potential progress that has been made over time. And so as you get more and more and better data collection, you can look at different ways to look at how have we improved over time. And that's really, really clear when you've improved patient outcomes by having them live longer.
I mean, I would say, I think another piece is sponsors will innovate if there's a signal that the agency who will respond to the innovation they're bringing in a positive way. And I think through the leadership in OCE, public meetings, activities through Friends of Cancer Research, longevity, FDA meetings, it's been clear that this is something that we all know if we aggregate this empirical evidence, we can bend the time to market on these drugs for patients. We can have more generalizable and broader data sets and more diverse than may be collected in the clinical trial setting. And so I think by the public-private partnerships regarding standards and how endpoints are defined that I know, John, you've been involved in as well, it created this perfect framework for this idea that we all knew would accelerate development, and then having that leadership from the agency really helps industry see, if you invest and prioritize in this, the partnership we have is it's gonna benefit everyone. Because really, what's our goal? Our goal is to get better treatments to patients faster, to have more treatments available due to the heterogeneity of individuals, and to have better evidence around efficacy and safety. And so to me, the advancement in RWE hits all those buckets. And so I do think having the FDA and the OCE framework, and it was the same around patient outcomes, really creates this great environment for collaboration and partnership. And I also think something I've seen across the board from OCE and patient outcomes is just the willingness to be part of academic collaborations like Sysaqol, Friends, Longevity. When we're able to bring a topic together like comparative tolerability, Jeff, I think is a great example, and have engagement across academia and industry and FDA around how do we address this need to better frame tolerability, it puts us in a really good position to standardize the evidence collection. And so if you were to say to me, what's the main analog rare disease could have, it's that the more we partner pre-competitively, align around core data standards, and partner, we can vastly accelerate drug development, which I think is to the benefit, of all patients and researchers and the agency.
Don, having been obviously inside FDA for a while these changes were transpiring and while FDA leadership was really helping push the field forward, I'm just curious your perspectives on this, in particular now having moved over to the pharmaceutical industry side.
Yep, thanks, John. I'm glad my video is working. Yeah, I think what makes perhaps the FDA oncology somewhat unique, besides all the external factors that we talked about, the revolution and molecular biology and sequencing and more rational drug development and immunology, et cetera. From an intrinsic factor, I think just having the leadership there that's willing to take risks, that's willing to engage externally with other stakeholders, the academic community, patient advocacy community, the professional societies, the industry. I think that's critical, and also just building sort of a critical mass of oncologists and other scientists who are, passionate about bringing new treatments to patients with cancer. So bringing forward that critical mass in the academic environment, to want to collaborate to solve critical questions. I think that was a recipe for success.
Yep. Yeah, absolutely. And I think one of the things that's interesting about this, this collection of people on the call right now is that we've all touched the regulatory use of real-world evidence in oncology in some capacity, whether on the pharmaceutical side, on the side of working to get industry partners together to develop standards, and on the side of actually developing those frameworks while at the FDA. But it takes a while to get there, right? We've all been working at this for a long time before we're kind of at this magical panacea of being able to use real world evidence in some capacity in a regulatory setting. So I'd like to spend a little bit of time talking about maybe some specific lessons learned about the steps that we've all taken to get to the point where the regulators and the FDA are using real world evidence. So maybe just talking about, and maybe Chris, I'll call on you to start. We talk a little bit about how we've thought about, and how in particular Amgen has thought about the data standards required for using real world data in regulatory settings. So things like what types of data do we need? How do we think about collection? How do we think about quality? And maybe let's just start there to try to tease out some lessons for the rare disease community.
Yeah. I think the real critical thing there is where do you kind of start? Because it's really a disease-driven question. For every disease, you need to figure out, first of all, if you're going to be investigating a specific population, what's the most appropriate setting to find those patients? Every data set that might be kind of naturally collected in the background through an EHR has certain biases associated with it. If you do a chart review study, that has certain biases associated with it. So always being very keen on knowing what are the limitations of the data you have. Is that data then going to be utilized, usable for that specific purpose you're trying to use it for, whether it's comparative effectiveness, whether it's safety signal detection, making sure all those characteristics are identified is really, really critical. Often, prospective studies probably give you really, really good information, but you have to be able to link that prospective data collection with the completeness that you get from something such as an EHR or a claims insurance database system. Maybe you need to link to those data to get the full picture of what's going on with those particular patients, especially in the instance of cancer where an adverse event that you're looking for may only happen during a visit to the ED where an emergency pops up. You can't really find those types of events otherwise. And, if you can't develop a framework that captures all of that, you're going to have incomplete data, and you have to design around that. And this is where the importance of having very thoughtful study design, having a good team work with you on capturing all the relevant data elements is really critical.
Yep, absolutely. And you kind of
touched themselves.
Oh, go ahead. Sorry, your video broke up for a second. Go ahead, Chris.
Oh, sorry. What I'm saying is, even though I can't speak to the actual, data monitoring side or the data check side, what you can say is that there's always going to be confounders, things that you don't know about whenever you're doing a study, and just making sure you can collect as much data as possible, particularly making sure all the published literature that are prognostic covariates that are known to have an impact upon your outcomes absolutely must be collected in all your data studies. So making sure that all of that is captured is really the most critical thing.
Yeah, absolutely. And one of the things that you're alluding to, which I think is a defining aspect of the progress in oncology, is really starting from that use case that you're trying to accomplish, so the study you're trying to do, the therapeutic program that you're attempting to push forward, the quality of life indicator that you're trying to measure, rather than just kind of going and saying, well, we need to build up this big data asset, let's just go and build something. So I'm wondering, Allison or Gidan, if you want to comment a little bit about how organizations are approaching that focus in terms of understanding that use case or that end utilization of the data, and then kind of building from there.
I mean, I can jump in. Sure, I'll go first. I really like what Chris was sharing, because I think one of the challenges with real world data is someone might say they've got, 2 million patients worth of data, and they might have, admit discharge records or, a lab value, right? And so it's not just about the number, it's about the depth and the richness of the record of the patient. And if the patient has a referral out of the primary oncologist to a pulmonologist for a respiratory event, or to a radiation oncologist, if you don't have that information, then the missed events you're giving introduce bias into any of the conclusions that you're providing. And so I do think considering the comprehensiveness of the evidence source is important. And that's in some way why I think standards are so important of what's collected and what's included in the analyses, so that you could, for rare disease, for consideration, you don't have to have one provider that has the empirical evidence you need. If you know you need information at the acute, subacute, and follow-up period of treatment, and you need a few core variables to draw meaningful conclusions, then you can aggregate that from multiple data sources. But I do think it's really important to remember that in oncology, missed information introduces bias, right? So if you're missing progression events, you can make a drug look more effective than it is, and it could just be related to the record keeping. But it's also that depth and breadth and comprehensiveness. And so when evaluating a real world data source, you really want to know the richness of the data, not just the number of the patients that they have. And if, as someone in industry, I'm consistently approached by providers of data. And so that would be my recommendation to anyone on the call wanting to really apply this in rare disease, is to really look closely at the quality, the depth, the richness, and the longitudinality of that information, or else all the great examples Chris just gave won't be feasible.
Don?
Yeah, totally agree with Allison and Chris. I mean, you know, I think the easier data to get at is the, you know, the structured data, you know, the lab values, the vital signs, et cetera. But a lot of the richness and nuance and complexity can come from unstructured data. And, you know, given that our healthcare systems are fragmented, you know, you have to be very much attuned to, you know, capturing all of the different care settings that a patient may drop in and out of longitudinally over time. You know, I think companies like SciAps and Flatiron and others have done a great job in, you know, curating unstructured data sets and creating, you know, very high quality real world data. You know, I think the work that needs to be done, and a lot of groups are looking at doing this, is to, you know, create, you know, standard data variables and collection up front, so that the back end collection becomes less intensive and onerous.
Yep, absolutely. So let's talk a little bit about one of the major issues, which is real world endpoints, and this is something that we've all spent a lot of time on. And you know, for those on the webinar who are newer to this area, you know, there's been a fairly consistent debate, working series over the past few years in oncology in terms of trying to understand the endpoints that can be developed and used from real world data, and the validity of those endpoints in comparison to the standard clinical trial endpoints that we're all familiar with. So I'm wondering if we could just spend a few minutes talking about how we've thought about real-world endpoints and development and validation. Maybe Jeff, Gidan, maybe starting with you, with both of you, and talk about some of the efforts from FDA, OCE, as well as friends on this front. Jeff, do you want to go ahead?
All right, I can start. You know, I think one of the reasons that we wanted to take the approach that we did by bringing multiple different data sets from multiple different sources together was really to just try and pressure test and see what we would come up with, frankly. You know, I can imagine, you know, and I'm saying this having never worked at FDA, but I could imagine that if your job is to review an application in front of you and a sponsor brings brings their application in with heavy reliance on a real world endpoint, for example, that has not been necessarily validated in numerous settings, and there's an approval decision on the line, well, then all of the issues that have been described around missingness of data, bias, you know, all of the other contributing factors become all that much more heightened because you're trying to sort of solve an unknown problem and, you know, weigh the quality of that information versus what you do and don't know for that particular scenario. So what we tried to do was sort of revisit all of those issues, but in almost a known situation. You know, so the scenario of looking at non-small cell lung cancer patients that were treated with a checkpoint inhibitor containing regimen versus chemotherapy alone was really not meant to try and adjudicate the clinical trials that had already been done, but the medical oncology community has accepted that the large randomized phase 3 trials that demonstrated the superiority of IO therapy over chemotherapy alone is sufficient to change the practice of how certain cancers are cared for. And so while that wasn't for debate, it created more of a neutral ground, so to speak, or a laboratory where people could come together and sort of pressure test what variables may contribute to differences in outcomes, what what different characteristics of the data may need to be examined further, just as sort of a non-controversial space to try and learn about the potential of these different endpoints a little bit better. And that takes a little bit of time. I'll be honest, we have gotten questions over the years of why are you trying to answer an already answered question, and that's fair. But I think that, while I'm not an incrementalist by nature, I think that is important to, take a stepwise approach in some instances rather than just a leap of faith. You might actually get further a little faster.
It's funny to reflect on that feedback because I would never kind of describe some of these efforts as kind of proving the thing that that people have worked on before. When you kind of reflect on a lot of these collaborative efforts that have been done, it's really not just the first time that organizations have done this together, but in many cases it's the first time that any of the individual organizations have done this to begin with, and then they're stepping up and doing it in this collaborative, somewhat awkward, setting where you're, revealing information to potential competitors before you've been able to sanitize it, and you kind of have to, get over this like semi-awkward dance that you have to do. So it's funny to hear that feedback because I'll say to everyone on the phone, the opposite side from the inside, it's not like that at all. It's actually it's very risky. And Gidan, as you're thinking about, if you're kind of putting the FDA hat back on, when we were all talking about this years ago, I imagine that there's this tension between you want to kind of push the field forward and you kind of have ideas in mind, but you also, might not think it's the position of the FDA to kind of push things forward, and rather you may want to be in a position to react to what folks are bringing you. So how did you walk that line, and how did you guys kind of come to the perspective of actually doing some of the studies that you did around time to next treatment and time to treatment discontinuation? Because you put a lot of effort into really putting a perspective out there for folks to react to, which, again, just saying from the outside, was really helpful in prompting organizations to move forward and not just sort of spin on concepts.
Yeah. Oh, thanks, John. I mean, I think, I think you described the kind of dichotomy well. I mean, on the one hand, we were very enthusiastic, and we even had a mandate from Congress through 21st Century Cures to better incorporate real-world evidence into our benefit-risk decision-making. But at the same time, to quote Dr. Pazner, we didn't want to preside over the death knell of the randomized controlled trial. So you got to always, strike that balance. I think that efforts that, Friends undertook, like the real-world endpoints effort, the tumor mutational burden harmonization effort, these pre-competitive kind of frenemy exercises are very important, to get everyone at the table, to come with, standard definitions. I mean, we put out a paper, looking at clinical trials data sets, looking at, time to treatment discontinuation with targeted therapies in lung cancer. And we found some interesting correlations between time to treatment discontinuation and overall survival. With some targeted therapies, which are, really effective, like ALK inhibitors, EGFR inhibitors, you might even give patients, treatment even beyond, conventional progression. So we were able to describe that. But what the Friends effort did was kind of standardize these definitions. Like I remember at the first meeting, one of the definitions for time to next treatment, the median time to next treatment surpassed the median overall survival, which was probably a censoring issue. But clearly, these weren't zombies living past survival. So I think kind of getting everyone at the same table, really rigorously discussing, what are the definitions, what are the best practices for defining real world endpoints has been enormously helpful for the community.
I mean, one thing, John, I think we might want to build on is I absolutely think the work that all of you are involved in is enormously helpful as we're advancing real world endpoints. In thinking about rare disease or even oncology, the FDA defines clinical benefit as a treatment improving how a patient feels, functions, or survives. So if we're only looking at part of that information, and in rare disease, where we tend to be looking at a more chronic illness, right, rather than a curative setting, which is what our intent and our goal is in cancer, then I do think, given the patient burden of a number of rare diseases on how patients feel and function, the patient experience data does need to be integrated with the clinical outcomes, or else you're only observing a picture of how that treatment or therapy is having a positive or negative effect on that patient's life. And so I do think for rare disease in particular, if the aggregation of real world evidence really will need to include empirical evidence on how treatments benefit, how patients feel and function in the construction of those patient relevant endpoints, or else we'll be missing a lot of valid information on endpoints. Like if you think about MS, right? We've started to make a lot of improvement in MS because we've got better endpoints so we can then understand the efficacy of those treatments. And so I think any type of standard, I know, Jeff, I was involved in some work with Laura from your team on this around cell and gene therapies and identifying what are the concepts for cell and gene therapies to be measured in the acute, subacute, and long-term setting to know how these treatments are impacting how patients feel and function. And so I think any way we can standardize that for rare disease as well with the incorporation into secondary data use sources, I think is going to be critical for advancement.
Yeah, absolutely. And in some ways our job, Chris, go ahead. Sorry, Chris, go.
So I think the last thing I just wanted to kind of throw in there too was I think because real world evidence and these types of non-clinical trial studies are still kind of in a very evolving space. I think it's really incumbent upon all of us who are designing some of these studies and putting new data out there that we look at what are some of the surrogate endpoints that we would observe within a trial, like an internal validity of it, and then compare that to the endpoint that we know is reliable within the data. So like if you want to look at time to next treatment discontinuation, see what that correlation is with the overall survival within that study. And if you could show the same directionality of that correlation with what's observed in clinical trials, that gives more and more evidence to show that these are very valid endpoints. So even though you may not be able to compare them externally between a clinical trial and some real world data, you can still say that this endpoint is valid, is a surrogate for survival in this particular setting.
Yeah. And, in some ways, like our job was a little bit easier than in the rare disease community in that we had pretty good endpoints, and job of real world data at first was basically figuring out, can we replicate those endpoints in a way that's valid and reproducible and trusted by the regulators. But, Allison, I think, and Chris, your comments about, how we move into better endpoint development are apt. So how have we thought about, and Allison, you've done a ton of work here, how have you thought about creating new endpoints using real-world data in oncology, particularly focused on the patient experience, and are there lessons that we can take from that sort of work into the rare disease community?
So it's a great question, John. From my perspective, I think we've aligned in oncology around what the salient core concepts are for patients. Tolerability of treatment, how that treatment may peak and ameliorate over the time on therapy, physical functioning, and then core symptoms of disease. So in highly symptomatic tumors such as non-small cell lung cancer, head cancer, gastric cancer, focusing on those concepts that can be shown to benefit from therapy. I think where there's still work to be done is our direct patient outcomes giving us the most valid and reliable data in the most seamless way for the patient. And so when I think about digital therapeutics, the potential is to better triangulate on those concepts and data sources using multiple modalities of data. And so now the approach we're taking is focusing on, combining digital symptoms with device data where it makes sense and it's an FDA cleared device and we have an endpoint. I'm not a big fan of putting in a device if we're not thinking about it in a clever way, that it can be integrated into the patient's life, and then aggregating that with risk factor data or impact data we can get from real world evidence. And so to me, the future of endpoint development is really kind of taking all these tools we've had in our kit that have kind of been separate, right? We've kind of had real world evidence, some digital symptoms, and some different types of device data. But now to me, it's aggregating that to get a more comprehensive picture of how a patient's impacted on a more timely manner, to understand those fluctuations, and then to aggregate those into complex models to then understand, and I, of course, am very focused right now on applying software as a medical device, right, around some of these endpoints to understand how to provide clinical decision support. But I think that's the place where I could see a lot of potential for rare disease, because the more comprehensive our assessment is of patients, the closer we're going to get to the signal, and the better we're going to be able to develop drugs or treatments or interventions or digital therapeutics that either improve that signal, right, if it's an efficacy signal, or ameliorate it if it's a safety signal. But I think that's the direction I see the natural next step of this. And what I think is really exciting about rare disease is you have so many areas where how patients feel, function, and survive is so critical, and you have a really engaged patient population that standardizing what gets assessed how and when, and then really crowdsourcing the analytics around those models, to me, has huge potential to advance endpoints.
One of the areas that I wanted to talk a little bit about as well is this move that we've made from cancers as kind of tumor type specific diseases, which are basically like mass collections of breast cancer, colon cancer, et cetera. We've made this move towards cancers as rare diseases, and in particular, molecularly driven or genomically driven. And I talked in the opening comments about a lot of the efforts from TCGA, and obviously, Jeff, there's lung map. But how can we help advise the rare disease community on the lessons learned from oncology in this move over the past few years? And in particular, how can we use the real world evidence domain to really help in advancing the understanding of rare diseases? So I'll toss it out to anyone who wants to start on that big issue.
I think I can maybe contribute a little bit there. I'm thinking of examples from the Blincyto program, where acute lymphoblastic leukemia was the first disease in question, ALL for short. It's a fairly rare disease. In the US, there's only about roughly 6,000 patients that are newly diagnosed with it on an annual basis. Half of that, roughly a little over half of that being pediatric patients and another half being elderly patients. What made our access to patients so feasible there was that we worked very closely with a cooperative group in Europe that helped to lead some of the patient recruitment, some of the historical data that we gathered to serve as our external control for that initial approval. And without the pivotal role of having a cooperative group like that participate with us in that data collection, there's no way we would have been able to have a successful study collection, data collection to understand, what this patient population's outcomes would look like and have a decent enough data package to file on that data and then show, look, this is a good, nice improvement over the standard of care therapies. I think that's particularly true of childhood pediatric cancers as well. And so there is a cooperative group mechanism that's already existing there. Leveraging those types of relationships is absolutely critical, getting all the centers on board, because then you can have as representative of a sample for collecting that type of patient data. And then you can have a nice, robust database, registry, what have you. Just find some sponsor that's willing to put the money up to help make that effort happen.
I could chime in. So as you mentioned, John, over the last decade, there have now been 4 tissue agnostic approvals in oncology, 2 with NTRK inhibitors. This targets a driver oncogene that's very rare in some common cancers, like 0.1% in non-small cell lung cancer, but common NTRK fusions in some very rare cancers like infantile fibrosarcoma. Very impressive response rates, which are consistent across the board, something like 60, 70%, very durable, some complete responses, really amazing outcomes. And then actually Merck has 2 tissue agnostic approvals, one for MSI high solid advanced refractory solid tumors, and the other is, as we alluded to earlier, a tumor mutational burden high advanced solid tumors. There you're taking advantage of the fact that some cancers, have very high tumor loads, high neoantigen presentations, a lot of T cell infiltration and PD-1 blockade, really irrespective of tumor of origin, has very robust and durable, long-standing responses. What can we learn, or what can other rare diseases learn from this approach? I think this really illustrates kind of the innovation and flexibility from the FDA Oncology Center. I think some other major health authorities across the world, haven't been as receptive because it is a change in mindset, if you will. From your traditional oncology drug development approach, looking at tumor of origin. But I think if you believe that the biomarker can select a unique subset of patients, and you see, really remarkable responses which are durable, across a number of different tumor types. That's what helped, bring forward the accelerated approvals for in these examples. One could imagine similarly, in non-cancer settings where there's, a driver, for example, molecular feature across several different disease states, and if you have a drug that's efficacious, across those different disease states, perhaps, a similar approach could be taken.
Yep, absolutely. Jeff or Allison, do you want to add anything onto this one before we wrap?
Yeah, well, I think, building on Gidon's point there, I think it's, in many ways that's sort of, how we've tried to choose some of these areas for pilot projects. Back to the idea of, they are essentially less controversial questions as opposed to, ones that you don't necessarily know the answer to. And I think one of the things that's hard in trying to define hard and fast rules that guide, successful drug development, at times it is up to interpretation. So you hear people sort of, at the end of the day, sort of say, well, you know it when you see it. So by looking at some of these non-controversial case studies, if you know it when you see it, in terms of like, around this question around real world endpoints, if something like time to treatment discontinuation or time to next treatment appears convincing in this prototype around non-small cell lung cancer, maybe when you see it in another setting on a drug that doesn't have as much information behind it, or if you're comparing it to a historical control or whatever the case may be, maybe you'll know what success looks like a little bit better. And oftentimes for a number of these rare conditions or under-researched types of cancer, for example, you may not know what success looks like quite yet. So can you transpose that appearance of success into other scenarios if you've sort of seen it in other situations as well and feel more confident about it?
Yep, absolutely. So I think with that, we are in fact out/over on our time. So unfortunately, we have to wrap up this session. So I want to thank everyone for attending today's session. Hopefully you found it informative to hear from the past 10 years of lessons learned and progress in oncology real-world evidence. Some of the key takeaways that I had from the session are the need to focus on defining and validating real-world endpoints, the need to understand how you apply subsegmentation to your populations, particularly as you look at molecularly driven phenotypes, and also the role of the FDA and collaborative organizations such as Forensic Cancer Research in helping to push the field forward. So I want to thank everyone for attending today. Thank you to the panelists for taking the time to participate, and hope you have a great rest of your day.
Thanks.